we can start shortly. Nivaldo, that overlapped with you in Lisbon, he unfortunately could not be here today. He has had some previous engagements.
I realize I have many Portuguese/Brazilian connections. It's cool.
Yeah, it's amazing how connected we are. It's a small world for sure. Okay.
Sorry, I'm back. Yeah.
Yes. Yeah. Okay. So we are live on YouTube already. So let's just, if you don't mind, Alex, let's just start. And then
share my screen.
Let me just create a brief introduction so the video stays there. So first of all, welcome to our seminar series. Thank you so much for participating on the seminar. We really appreciate, and it's a pleasure to have somebody with such a breadth of work on behavior. Alex is from Spain, from the Instituto de Neurociencias de Alicante, Spain. He did his PhD in University of Barcelona, and then did a couple postdoctoral in Portugal, right? And another one in Spain as well, right? Yes. And you originally you're a physicist, correct?
Yes, a theoretical physicist, pen and paper for 4 years before turning into the real deal of biology. Yes.
Fantastic. Well, thank you so much. It's a great pleasure to have you, and the floor is yours, or the screen is yours, I should say.
All right, let's see. Host disabled participant sharing screen. Yeah, allow it and I'll do it. Yeah. All right. Here it is. Okay. Okay, so you let me know if there's some weird things with my screen. Otherwise, I'll assume you can see the first slide, right? Yes. All right. Yes. So it's weird to give a talk like this, but it would be worse to do it like that. So in a way, that's fine. Okay. So yes, I'm at the Instituto de Neurociencias in Alicante. I lead the Behavior of Organisms lab. I'm a theoretical physicist by training who turned into neuroscience, and I'm really fascinated by behavior. And as you'll see, my approach is mostly theoretical, but that does not mean that I spend my time in the whiteboard necessarily. We always analyze real data, or just data, but we're trying to abstract away and think of principles of animal behavior and across species, although today I'll talk to you about C. elegans. Okay, so let me start by motivating, and not to be polemic, just a bit, but by motivating the talk by this quote by Richard Dawkins. Before he became the Richard Dawkins we know, he was a really good ethologist. And so he writes, if we look far into the future of our science, what will it mean to say we understand the mechanism of behavior? The obvious answer is what may be called the neurophysiologist's nirvana. Complete wiring diagram, blah, blah, blah. So of course, there's nothing against it in principle, but what would that nirvana really bring us? And then he says, well, real understanding will only come from the distillation of general principles at a higher level. So really that's what we are after. Another way to put it, also metaphorically, but technically, is through Mars 3 levels, right? And sure, we want to know how things are implemented. That'll be here level 3, right? But really to make sense of it, we should also look for the rules, the algorithmic version, if you want, and the computational, what's in that case, the animal trying to solve. And again, these things make sense, but it's often hard to really cover the 3 levels. And sometimes if you're really concentrated on one level, you may forget that another level is really necessary to make sense of the whole story. So this birds analogy is quite nice, right? Like, of course, we want to know the properties of feathers, but neuroscience sometimes, it's, maybe that sounds weird, but it's too obsessed with neurons and maybe too little obsessed with behavior. And I think we need to care more about behavior, because the main reason why we have a brain is to act. Now we wrote some of these ideas in a lovely piece that became quite popular, at least unexpectedly to me. If you haven't read it, I recommend this piece by Krakauer and Gazanfar, McIver and Popple and myself, where it's essentially a plea for behavior in neuroscience. And this falls into this category of things that are like obvious but non-trivial. Because I think everything we're saying here is quite obvious if you think about it, but then when you look at whether it's being taken care of, most of the time it's not, or many times it is not, so it's non-trivial. And 2 years later, we wrote, Asif and I wrote this kind of follow-up piece where we said, sure, neuroscience needs behavior, but what do we really mean by behavior? And just to go a bit to walk you through this figure, but again, I recommend you read it if you enjoy it. Many times we think of the brain as if it was floating in a vat here. Do you see my cursor moving, or do you don't? You do, okay, as if it was in a vat here, but really, and again, obvious but non-trivial, the brain has a body, as the famous paper also claims, and it's living in a world. So let's think of animals as animals, not as a brain carried out by a bag of muscles, if you wish. Then we have this tendency of thinking of behavior as merely some sort of output of a response in a sort of reflex arc, but really animals are in closed loop. And again, this calls for a less anthropocentric view of animal behavior, where really what animals do is not just to produce output, but actually to produce output in order to create certain consequences that they can perceive and that they wish to. So yes, behavior is more a control loop than a reflex arc. And also, come on, animals have purposes, we have purposes. Well, this is a philosophical issue, but and also a theological issue, but it's really weird for me to study living beings as if they were machines. Now, another principle that we articulated here is that because of this input-output paradigm, when we observe, and we do observe variable behavior, we try to tame it with statistics, and that makes sense. But we should also take into account that not from the statistician point of view, but from the animal's point of view, different means is a good way to achieve the same goal. And I always put this stupid example, like if I'm if I'm cold in the room, I could control the temperature by either putting a jumper on or turning the heating on, right? So these would be very different means to achieve the same goal. Now, from the scientists outside, you perhaps would, maybe with a very sophisticated behavioral analysis tool, classify these as different behaviors. But from the organism perspective, in this case me, it will be the same behavior. And also, and this is really hard for neuroscience because we're really focused on here now, but development plays a very important role, and also evolution, right? So expanding timescales, it's also a challenge. And now, well, let's get into the worms soon and what I want to present, but one more slide. So really, I've been also trying to articulate this question, which I think is vital in neuroscience, which is, again, anthropomorphism, from whose point of view we're studying. And I love this caricature by Steed, where you see these guys drawing what they see, which is this skull, but the tree is drawing the wooden part. All right, so in the lab, we study principles of animal behavior across species. We try to establish principles, and we do so by merging hardcore, I would say, that's my bias, conceptual theoretical work with experimental data, and computational analysis is what helps us bridge these two. Now, the things that I do in my lab is quite heterodox and often eclectic. So we work on causality. All this works with C. For some reason, they all go with C, causality, and exploring what counterfactuals can really tell us about the relationship between neurons and behavior, but also control, and where I was talking about the feedback and circular causality and constraints, complexity and coarse graining, which is what I'll talk about, and also context, or really what we call real-world neuroscience. All right, so I use these triangles to illustrate that we use different species and even robots, if you consider them species, different let's say model organisms to tackle different questions that we are interested. So here on the far right, this is somewhere you can check in our website. We study magic with magicians in the real world to study human cognition. Now we do some work with robots to better understand the embodiment of the algorithms that we designed to accomplish some bio-inspired tasks. And today I'll talk to you about worms, which really are a great organism to do what's called physics of behavior, and which is another way of saying some physicists, transition to neuroscience or ethology, and we want to do things very precisely and be able to have a lot of good data to throw at it, concepts, and the worm is really a great organism for that. So I know neuroscience is quite rodent-centric, that's fine, but bear in mind that worms are really powerful also. So this is our lovely creature, right? And let me first tell you about our attempts to establish hierarchical organization as a principle of behavior in worms. And we started this by this simple idea. So you could have here a sequence of letters, A, B, A, B, A, B, A, B, or this one here in red, right? Now, there's something called the minimum description length principle, which says that the more you can compress something, the more you understand it, or the better it is explained. And of course, this can be questioned, but in any case, we can apply this very readily here, because instead of saying A, B, A, B, A, B, I just need to say A, B repeated n times, right? Whereas this one here is quite uncompressible, totally uncompressible, actually. So I could try to estimate behavioral complexity by compressing data, right? Now, what data are we going to compress? So we're using this really amazing data set, which was published in 2013. It's more than 1,000 N2, which is the wild-type worm individuals, more than 1,000 individuals, and there are also mutants and wild strains and so on. And what description are we going to use? And we'll talk about descriptions a bit more at length later. This one, where the body of the worm, the posture of the worm is discretized into 90 postural templates. It is as if you would now transform the continuous body posture and movement of the worm into discrete symbols or letters. And now, as you see down here, the dynamics, it's going to be, well, now template 11, now template 10, 3, 75, so on, right? So this is going to be very useful, as you'll see. And again, you could use other descriptors. And in fact, what I'm leading to with this talk is that in neuroscience, I would say, and in behavior especially, we don't know really what we should measure. It's more like the Wild West. We measure what we can, we measure what we want. If you study rodents, and somebody wrote like 10 papers before saying you count how much time it spends on a corner of a square box, y squared, well, in a square box, and then you count for 5 minutes the amount of time that it's there, that's your behavioral index. But if you study another organism or even the same mouse, you would use another behavioral descriptor, right? So in physics, we're spoiled. We know what we need to measure when we're studying, let's say, mechanics, and there's this rock, moving through space. We know, we need to measure position and velocity, and we know that what's relevant for force is the second derivative. Or you could say similarly, although it's more complex than that. When you measure neural activity, of course, you need to concentrate on spikes and so on. But in behavior, I would claim we have no idea. So everybody does what they want or what they can. Now, having said that, this description is quite useful because here we have worm behavior. This is an actual track of a worm, and just crawling. It could be in the presence of food or in its absence. So this is the trajectory. Now we zoom in to the posture, and now from now on, we're gonna substitute the real posture with the template the best match. And it's important to say that in this previous publication that I mentioned, they show that with 90 templates, you could cover a great deal of the variability. So now, for instance, crawling forward for the worm, it's going to be 62, 11, 64, 19, 72, and so on, and the duration. So here you see how you can represent 1 animal, 2 animals, 25, 50. Here I'm plotting 250 animals as a function of the postural sequence. So, well, depending who you talk to, this is big data, but in any case, even if it's not big data, it will allow us to computationally estimate some interesting quantities, let's say, but this postural template representation. So the first thing we did is to try to extract the structure or the organization of behavior of the worm by compressing hierarchically those postural sequences. And so this is what the algorithm does. So they could have a sequence 1, 2, 1, 1, 2, 1, 2, 2, etc. And you could ask, what's the most compressive motif, the most frequent, or the longest? Right? They could be interesting for different reasons. Now, if you ask for the most compressive, it turns out to be, in this case, it's 1 to 1. So you would now design a new rule that says every time I find 1 to 1, I'll call this 3. Now my sequence is shorter, and I recursively apply this until I cannot compress anymore. So to make a long story short, what this allows is, first, you can reveal the structure of your data as compared to some models like a Markov model or a more deterministic model, because you can ask, how much can I squeeze? How much can I compress my worm data? And this is what we did here. So it could be a measure of phenotyping. Another thing is that as you compress this, you get you get these trees, right? Because you're recursively compressing this. So you get these behavioral motifs, and then you can look back at them and see if they make sense biologically, and they do. So these are automatically detected motifs. And this has to do with reversals, pauses, and turns. And also, as I was saying, you can use it to phenotype mutants. Okay, let me speed up a little bit. So if you do that on text, real text, human novels, you could see that it more or less works. So it, put together th and is here. So you have this word "these," which is well captured here. The word "tablet," okay, but there are of course some errors here. So this M of memory doesn't go with the word memory, so it goes here. So that's just to remind you that we're trying to pull out syntax, but of course we know nothing about meaning in the case of the worm, nor in the case of just running this algorithm through human text. Another limitation is that we started with a sequence and we are extracting hierarchical organization via compression, but what we would like to have is something that can also generate the behavior from the top to the bottom. So we would have here this module D that is comprised of these 3 modules C, and then in a way like the studies of human syntax and human language syntax, we could generate behavior. So for that, we moved to the idea of hierarchy of content, where you are lumping things inside things, and we did this through compressibility, as you just saw, to a hierarchy of command. And how do we do that? We do that through the notion of replaceability. So let me briefly tell you how this works. So this is work of Saurabh Gupta, and in essence, we're inspired by near decomposability and by these 2 landmark papers, I would say, by Simon and Dawkins. So the idea is, rather than looking for co-occurrence of postures here, of worm postures, we are going to think in terms of the menu, right? So if you would want to make groups of meals, of courses, it's hard. You would not find spaghetti close to paella, because they are the second course. So what you need to look at is whether spaghetti and paella sometimes have before salad and after, let's say, the third course or dessert. Of course, this analogy can be bent by saying, well, I could ask the waiter to have this too, but that's not the point. The point is that by looking for mutually replaceable elements, we can find groups that would naturally, and that's what we hope, would naturally expose a grammar, in this case for C elegans. So this is what happens when you use mutual replaceability, and you find that the 90 postures with which we started, let's call this the microscopic description. You can find 10 clusters or chunks of mesoscopic description, which in turn group into 3 macroscopic clusters. And this is going to be important in a moment. Now, if we order now the 90 postures, and every time we found like microscopic posture 12 here of the worm, we ask to what submodule or module it belongs, then all of a sudden we see this clear structure in the transition matrix, which means that this is actually revealing order and capturing predictability, in this case in how the worm is roaming. So when the worm is moving forward, it's most of the time going from this submodule B1 to B2 and to B3 and so on. So this is a summary. So we would have roaming now composed of these 3 modules, which in turn are decomposing these 4, and from here you can choose. So this is quite cool because it means now you allow for flexibility. Let's say the worm will be choosing now posture 41, but then the dynamics, the hierarchical dynamics is, okay, now you need to move to submodule B2, so choose another one here. And that's how the worm, by going from left to right, or here like clockwise, as you see, it's moving forward. So this, although it could be a bit technical and we can discuss more, this allows me to enter into the problem where I wanted to arrive for this talk, which is that we have an animal, a worm or a mouse, whatever, and we measure it as good as we can. This is feasible and easy today. Now we need to choose a description. In this case, we chose a discrete description based on the posture. And now, even then, we could still decide to coarse grain this description or make groups and so on. And again, you can do whatever you can to be able to reveal structure or even predict differences or detect differences between mutants and so on, right? But then there is this question, and I leave this here blank so that I remember to drink some water. So again, there's no, to my knowledge, there's no what we could call theory of measurement in biology, or if you don't want to say it so broadly, at least in neuroscience, and when I say neuroscience, I mean neurons and behavior, but let's just say ethology if you want, because there's a lot of theoretical work going on in neuroscience if you want. In ethology, again, we don't really know what to measure, and it's also really hard to compare. Like if you're studying a fruit fly, you may know a lot about the fruit fly and even neural mechanisms and molecular components, but then the behaviors of that fruit fly rarely can be, transposed to a behavior of a rodent or a behavior of a primate, let's say. So in C. elegans, again, let's explore this. So here we have C. elegans. Yes?
Like, I was wondering just a small thing, like, was the time warping thing, was it like a no-brainer or was it painful? Like,
no, yeah, thanks. It's a very
sophisticated. Did it bring up issues, or?
It is a very sophisticated word to just say we're gonna just ignore the duration of each. So every time
I'm wondering not how you do it, but actually, like, was it without consequence? I mean, because it seems like time.
Yes. So there's a technical and a biological answer to it. So the technical is that if you're going to use, in that case, when I was describing, well, I won't go up, but if you're describing the compression algorithm, because we have quite good temporal resolution, if you don't time warp, means that you're going to have the worm in posture 12, but then next 166 milliseconds, you're going to have it at posture 12, and then maybe 12, and then maybe 12, and then maybe it will jump to 72. Now, if you don't time warp, then those 4 times 12, it's going to be selected often as most compressive, perhaps. And so you're gonna waste what your algorithm can do. So a trick is, okay, let's forget about time for a moment. Let's compress that. And then I didn't show it to you, but then you can bring time back and ask, well, so does compression, you can plot compression as a function of versus how fast the animal switches from one posture to another and so on. So it's like, for convenience, we take it out, but we can always bring it back in.
Okay.
Yeah. All right. Yeah. Thanks for interrupting if things are not clear or you want me to emphasize. Okay. So now we're at a good point, because a good point for really getting into the crux of the title, which was, I remind you, carving. You could say carving circuits and behavior at its joints. You know, this famous expression, carving nature at its joints, right? Okay, so here you have C. elegans, the tracks of C. elegans. You could say it's simply locomoting if you're more like let's say, a motor control guy, but it's also foraging and navigated. So if you're more a sensory guy or a internal state, whatever. So that's what it's doing. It's moving in this agar plate. And it's quite two-dimensional, so that's one other reason why physicists like it, because again, the posture is really tightly linked to its movement. So with the posture, you think you capture most of what the worm is doing. So there are other descriptions, other very good descriptions, the famous eigenworms by Stephens et al. So the other continuous, mostly continuous descriptions, but I'm going to continue hammering on this postural sequence that's discrete. So basically, again, treating worm data, behavioral data as text. So I showed you how you can extract compression motifs, but I pointed to you some of the limitations of that. I showed you briefly how we can find with replaceability these modules. But okay, then one can do two things. Well, one can just leave it at that. One can exploit much more these descriptions. One can look for other ways of partitioning, but just instead of partitioning in other ways just to explore, I became more concerned about what I call the problem of construction of observables, which is, again, a fancy name to say, well, I already mentioned the Wild West of ethology, where you decide more or less what you're going to measure, but there's another side of this coin, which is that every time you want to measure something, you are already coarse graining, you're already throwing information, you're deciding. So measurement is also, whether it's conscious or not, is an act of deciding what you're gonna put together and what you're gonna ignore. And we physicists know that, and I think biologists writ large also know it, but here I'm gonna try to make it explicit. So this is the problem, and we relate it here to a wider problem, which is replicability, just to put it in context, but you could think of it in a more theoretical way. But let's imagine replicability. So you know, this famous paper, most results, most biomedical results are false, and then the media bangs a lot on psychologists. That's something that neuroscience seem to like doing, banging on psychology, saying, well, the results are not replicable. But also, I would say generally, replicability is an issue, right? And how do people try to address it? When one way is before everything starts, you have an experimental design, maybe you standardize it, right? And of course, you are careful and so on. And so you could address it at the experimental level, or once you finish, you can be very cautious and fair with the statistical analysis you do. And maybe there'll be a third level, which is more like socio-political, which is changing the incentives that push many people to hack statistics and so on. But here's something in the middle, which we call the epistemic problem, which is that whatever you decide to measure, even before you apply the statistics, by necessity, you've already done what this square tries to represent, which is, this is what in physics we call the phase space. Any trajectory, you can conceptualize it as moving through a space, right? And here it's just two axes for simplicity. It could be position and velocity, it could be anything. And because you don't have infinite precision in time or space, basically you're going to say that now your time series is in this style, and now in this style, and now in this style. So you always need to decide a coarse graining. Now, microscopically, it turns out that different coarse grainings can lead to, the same coarse graining, the same microscopic state with different coarse graining can lead to different macroscopic states. And it could also be that different microscopic states go to the same macroscopic state. I'm sharing screen. Do you see? Yeah, you see what here. So let's try to illustrate this, the problematic with this, or what happens, basically what happens as you choose different coarse grainings. And again, I'm going to use the worm, and I'm going to choose certain ways of carving worm behavior at different joints, and then I'm going to explore what happens when you do that. And before I go with behavioral data here with the worm, I'll show it to you in a very nice and paradigmatic mathematical model, and also in a network simulation. Okay, so let's go with the maths here. And for that, we're going to use the tent map. So this slide summarizes it all. Let's go slowly through it. Let me see how I'm doing with time. I'm doing good. Okay. So the tent map is perhaps the simplest discrete mathematical mapping you can have, which basically says, well, so you just give it an initial value between 0 and 1, in this case 0.2, and then it's a function t that says now, okay, so now you start at 0.2, now you're going to go to 0.4. It just gives you the dynamics. Next point is 0.4. Okay, so now you're at 0.4, next one is 0.8. So basically this tent map is very well studied mathematically, also through some really technical aspects that are called what is it? Generating partitions, which has to do, for the more technically oriented part of the audience, has to do precisely with how we can make proper partitions. But in any case, the 10 map is going to generate a dynamic. So this 10 map here generates these dynamics. You see it here. So the microscopic dynamics is going to be these points jumping here, right? Now I'm here, now I'm here, now I'm here. Now, you could say, well, I'm going to coarse grain these dynamics, and I'm just going to, how do I coarse grain? I decide a partition here at this point D, and if it's above, I'll call it 1, and if it's below, I'll call it 0. It doesn't really matter how you call it. So here you make two macro states, above and below. Now, if you do that, now you can calculate, and this is going to be the mathematical object we use. You can calculate what we call predictive power, which is nothing but the mutual information between the current state and the future state. So basically, this tells you this is a measure of the information in bits that can be obtained about the future if you know the present. So it's, yeah.
Can I add something?
Yeah.
So the idea is that you have a 10th map model, I don't know if it's a family of models, but then you adjust you adjust your data through that kind of model. Like, what defines the shape of the behavior of the 10th?
Sure. I mean, this is, I'm not talking about the worm now. Sorry if it's confusing. Here, I just chose what's mathematically called the 10th map, which is a very particular mapping, which is literally this that's why it's called 10th map, because it looks like a 10. So it's this straight line with this slope up to 0.5 and then down. And this is what defines the dynamics in this case. We're going to go to the worm in a moment, but for now, we just need to know that we're using the 10 map. The 10 map generates the dynamics, and now we use this idea of predictive power or mutual information to, basically, we're going to estimate the mutual information of the present, of the future given the present for this data series of the 10 map, and later we'll do it for the worm.
Okay. Yeah.
Okay, so we cut. So if we choose a certain if we choose two partitions, we could be moving the partition right along this axis, and we could be asking, as we move the partition, how does this predictive power or mutual information change? And if we do so, we see that there's a peak here, a maximum here, minimum, which is actually 0, and a local maximum here. So again, to not be very, very detailed, this is telling you some relevant things. Like if you would decide to carve the 10 map at this joint here, your predictive power would go to 0. So it would be really bad. It means like you could not really guess what's coming next based on the state now. So microscopically, let me remind you always these levels. Microscopically, there's a dynamics ongoing, but you decide you're only going to look at 2 macroscopic states. And of course, you could say, no, okay, I will going to look at 3 and 4. But my point is that there's a finite limit to what you can look at. So if you would only look at 2, that would be a really bad choice here. That would not be bad, and that would be the best you could do for 2. That's the maximum predictive power if you get it here, if you would cut here. Now, if you say, okay, let's gonna keep this one here, this one here, this star, and let's make a third one. Now, if you explore like this, if this was a knob, different places to make this third cut, then you see, again, some interesting things, I think. You can still bring predictive power up by cutting here, right? But look, this is interesting. If you would be cutting to the right, you would not improve your predictive power. So again, this is a very simple model, but the message is measuring more, it's not always going to help you. I think in the era of big data craziness, that's relevant message despite being just with the 10 map. Like more detail here won't increase your predictive power, and that should be where you put it. So here in I have this, we have summarized this. One cut when we make one partition, when we make 2 partitions, and here you see from the microscopic dynamics, the macroscopic dynamics, how it would look like with 2 states or 3 states, right? So again, the 10 map helps to really understand that, because you could solve some things analytically, you can simulate, and this is the message. Depending how you're making these groups, you see that these different things can happen. Have no predictability. Doesn't matter if you add detail, or have much more predictability just by, you see, if you would cut here at 0.3 and something, you have much more predictability than if you do it here at 0.4. Okay, so let me now go to something a bit more realistic. It's not the 10 map. It's a simple network model. And then I'll go to the worm and I'll wrap it up. Okay, this is a very also a very crowded slide that I'll try to explain slowly. So stop me if something is not clear. So let's apply the same logic now to this stochastic network model simulation. So we choose 4 nodes, and the green arrows are activate, and the reddish ones inhibit. And these 4 arrows here, it's just the injection of some stochastic noise, either 0 or 1, and basically these nodes receive 0, 1, and if they receive 1 or more, then they're active again. So again, this is created on purpose actually to show you what you're going to see now. So don't ask me where this comes from. It's not really bio-inspired. It's just a very simple network that will make the point we want to make about the importance of choosing your partition, right? So now the node states can be on and off, 0, 1, that I just said. Now this is how we can make partitions. We could decide we're going to make these and 3 and 4, so we'd have that. Or we could decide we're going to lump together 1 and 2 and 3 and 4. It's this one here in red. Or we can decide we're going to lump together 1, 2, 3, and then 4. Or we're going to lump them all together, right? So you can basically enumerate all possible combinations of these partitions, right? And the values they would have, because they could be 0 or 1. So you could if you lump together 1 and 2, so the total value could be 0, 1, or 2, right? Now, in C here, you see all possible partitions again, like this, horizontal, vertical, crossed, and then these 4 edges. This looks like, by the way, I don't know if you have it there in Brazil, like IKEA, like IKEA instructions. But in any case, it's a bad joke. Okay, so these are all possible partitions.
We don't have it.
You don't. Yeah. So these are all possible partitions. Again, simplified. Remember, a moment ago, I was showing you just cutting in half the 10 map, cutting in 3 times, in 3 parts. Now, this is like all the ways you could cut these 4 networks, right? Now, if you have, so here you see examples of the microdynamics of these 4 nodes once you simulate them, and then some examples of what you would be seeing macroscopically if you choose partition I, E, or B, right? Looking at these is not so instructive, but what's instructive is when you, again, estimate numerically the predictive power through mutual information between the current state and the following state. So this tells you, if you do that for the, let's say, mostly resolved partition, right? This one here in blue, that's the predictive power you get. I mean, you could discuss what this means in bits and so on, but the point is here that if you use this coarse-grained case, this one here, right? For B, which is this one here, you get exactly the same. So this is telling you you can afford to lose detail. You'll still have the exact predictive power you have microscopically. So you can see already this would be a good place to carve the circuit that it joins. But now if you, instead of B, would choose C or D, this goes really down. Again, this example is selected it's intending to show that, right? So it matters. It matters how you're gonna, not only, this could be at the level of measuring or at the level of the analysis or pre-processing if you wish. It really matters here. Then for these 4, it's more or less the same. And of course, you would expect that if you just lump them all in one, this is just going down. Okay, so let's exploit a bit more this idea for this simple circuit and zoom out is for replicability, as I was mentioning before as an introduction. So now let's imagine that we have this system. Again, the system is this network of 4 nodes, and we can choose this coarse-graining one, which let's call it the good partition, or coarse-graining 2, which is, let's call it the poor partition. Remember, like the good, that could be a good one, and that could be a poor one because of this, right? Now we know they're good and bad. Now, the system could be in a different microscopic state, right? But because of the partition, 2 different laboratories would see it at the same mesoscopic state. And this is important. Like when you try to prepare your experiment, you would want to have the same initial condition. Of course, this is never possible microscopically, but macroscopically you could make an effort, or mesoscopically. So you could prepare it the same. Now, for the good partition, you would see that both labs see the same temporal evolution. So this is iterating 1 and 2 and then all the way to 10 steps, that model. Now for the bad partition, although you would maybe prescribe the protocol to do the experiment and set it up in this same initial state, you see here, that's the important here, different temporal evolution, and then it converges, and this is for another reason. But the point here is that choosing a partition, it's not just to have more predictive power, as I was illustrating here, but also it could lead to differences in the temporal evolution, even if you carefully prepare your system very well at the beginning, right? Okay.
I'm sorry, I didn't get it actually. Like the lab, what's the difference? So lab A and lab B?
Yeah, so lab A and lab B have different microscopic preparations, but they don't know because what you can measure is mesoscopic. So this is the system. This is the coarse graining that the scientist chooses. This is the microscopic preparation of two different labs. So it's a bit complex because of this. There are two partitions, two different types of partitions, same system, different microscopic preparations, and then different labs. So the point is that it matters which partition you're using, even if initially you would think that everything is the same mesoscopically. And you would see this through temporal evolution. And so perhaps, again, this is an idealized case with a simple model, but then this would lead to lack of replicability, because now another lab comes and they don't get the same temporal evolution. So if they would stop the experiment here, perhaps they wouldn't find the same results. Even if they would use, as I was trying to illustrate here, you could prepare it very well, you could do the stats very well, but it's just because of this epistemic choice that you made of your partitions, you're having trouble, not just with predictive power, but with replicability. All right, so to finish, let me show you this, and that was very important to me when we were doing that. And let me mention explicitly, this is work by Marina Vegué, a postdoc that was in the lab. So it was very important for me. Again, coming from physics, I'm always very careful not to try to explain the whole universe with a harmonic oscillator, right? Which is what we learned for 4 years. It was amazing, right? You come out of physics thinking you can understand everything, but then you come across a simple maggot or a worm and it doesn't make sense. So it was important that it's beautiful to use the tent map. It's beautiful to have this numerical simulation, but now can we run this on real data and also make sense of it and extract some lessons about how we should measure things? So yes, we can do it. And that's why I explained to you before this representation of worm behavior. So again, this is C. elegans behavior. This is the trajectory in XY space. This is its body and the head. And we, once more, use this discrete postural sequence. Now, before I explained to you when I was using mutual replaceability that you get naturally these 10 different modules, remember? And I told you that you can also lump them. The mutual replaceability algorithm lumps them into 3. So that's quite convenient now, because we can say that the 90 templates is the microscopic description, the 10 chunks is the mesoscopic description, and the 3 bigger chunks is the macroscopic description. So we have these 3 levels of detail here, and it's based on coarse graining as well. Now, let's explore 2 more ways of partitioning the data. So you could, instead of asking for mutual replaceability, this analogy of the menu and the spaghetti and the soup, you could simply say, well, let's lump together postures that are more similar, just morphologically. And that was also very important. I didn't mention it in the previous study, because we wanted to see if what we would get from mutual replaceability could not just be explained by, well, that the worm is in this posture, and of course to transition to another one, it's gonna explore just a set of similarly morphologically similar postures. So you can make different partitions, again, micro, meso, and macro, and also you could decide, well, overall, I'm gonna partition the worm. Well, if all these local angles are more to the left, to the right, or straight. And in a way, we do this all the time. I mean, I have experience with studying fruit flies and rodents as well. And I read it all the time, like you decide, well, the more or less, the mouse is bending to the left, it's bending to the right, it's more or less straight. And you could build 3 states and just do your neural business with it, right? So it's not just weird things. You could potentially decide these 3 ways of parsing or coarse graining your data, and in these 3 resolutions. So this is what we find now. If we again use mutual information as an estimate of predictive power. So let me walk you through this. So this is for the macro description and the meso. Remember, right? We always have 90, but we can arrange them in these 3 ways, and in these 3 ways, mesoscopically and macroscopically. So, and here you're going to see MR, mutual replaceability grouping, morphological, left and right, and then some random control. Like you will take the 90 template and just make groups, whatever. So macro means we're going to make 3. Meso means we're going to make 10, right? Now, for macro, this is telling you if you make groups of 3, in this case, it doesn't really matter which of these 3 partitions you use, because it doesn't matter here. This is locally optimal. So it's going to be like, I'm going to walk you through partition flips in a moment, but let's just imagine you take these partitions, these ones here, and you estimate mutual information. They are all here. Now what you can start to dissolve them and just randomly mutate postures from one group to another, from one group to another, and average. And then you will see that how many partition flips you need to make, so that on average, you converge as you should towards the random partition. Okay, there are a few flips you need to make, but the point of this plot here is that if you make 3 groups, it's quite coarse-grained. It doesn't really matter what you chose. They are all more or less lay in the same place. Now, if you go to the right here, if you make 10 groups, then this is giving you a criteria. This is telling you mutual replaceability is much better here, because you get much more predictive power. Actually, significantly much more predictive power. And then you would choose morphologically. And this left-right bending, it could give you some, but it's not very good, and here it's random. And again, as you flip them, they should all converge to partition flip. So the message would be, at this level of resolution, it doesn't matter. At this level of resolution, it starts to matter. Now, there's something that's important here, which is that, okay, here I'm making partition flips randomly and averaging. But what if I imagine the worst scenario, like the scientist, the experimenter, really has bad luck and makes these groups, but maybe flips a posture 82, instead of putting it here in group 2, puts it here in group 6. So if I just mutate one posture, right? So what's the worst possible choice I could make? So that's what you see here. This is again, predictive power. Now, if I choose to move posture 19 from group 2 to group 1, this drops here. And if I then choose to move posture 62 from group 1 to group 3, this goes here. And you could say, well, who cares about these postures? But the point is that I could be so unlucky that out of 90 templates, I just partition these 2, and I lose half of predictive power, right? So this goes back to that tent map plot that I was showing you here. Like I could be here, but I could slightly place my partition somewhere else. Just a little bit of error in the code or how I define it, and I would be losing a lot of predictive power. So this is what this panel here shows. And when you make 10 groups, it's also the case, but it's quite spectacular. I wasn't expecting that just by flipping 2, you could lose half of the predictive power. Okay, just not to bother you with that, we can look at the level of individuals. So each dot of this is an individual. Instead of pulling them together, I mean, we could do that because we had more than 1,000 worms, so we pull them, but we could try to estimate, again, this mutual information for different ways of partitioning, and each dot will be an individual, and we can look at certain things. Maybe we can look at the least predictable animals and the more predictable animals, and also how these differences change as we compare one partition versus the other, 3 groups, 10 groups. I mean, this is more details, but the story doesn't end here. Let me just zoom out, because I don't want to, again, be too technical here. Why I think this is interesting. So one thing is from a predictive point of view, and this is one of my favorite, I mean, I'm always struggling with this, what the scientist cares about, what the mouse cares about, kind of my plea for animal centrism in neuroscience. So sure, we could use this as a predictive tool. You're gonna numerically make different partitions, and you're gonna choose the one that has more predictive power. Good for you, you can predict better. Another, like a second level, would be understanding. So not just predicting, but going back and trying to understand, for instance, here, what's special about posture 19 or 62 that seems to be so important here, or why mutual replaceability is much more predictive than morphological similarity if if maybe you would think there's not such a big deal. So that would be understanding. But then there's a third aspect that I haven't worked out, and I was talking with Dan before we started live, that although I presented those perspective pieces in Neuron, right, in 2017 and 19, where I could say, we preach about how one should study behavior, it's often not so easy to preach with example, right? So the point here would be, that's very good, Alex, but that's just talking about me, the scientist, doing a better job. But for instance, here, from the point of view of the animal, why would that be important? And there's some nice quotes I won't read from, for instance, Schrödinger, What is Life, which says that, of course, animals, if they need to coarse grain too. If they would be responding to the microscopic laws, it would be a craziness. So they also guide themselves by microscopic laws. So what I'm trying to say here is that coarse graining is inescapable, both for the scientist studying the animal as for the animal. And maybe, and I'm just saying maybe, this is a way to try to establish a theory of measurement in biology. At least what we did here is be more conscious of the fact that epistemology, what you choose to measure, has an impact of what you find. It's not just what the data says, but you made many choices there. So just a summary slide, and more zooming out about our approach. Sometimes you have ground theories and you have no evidence or very little evidence. That's tempting to me, but I'm not so interested. Sometimes you have no hypothesis, but big data or necessity and sufficiency claims that I have criticized in the past. Well, you can sometimes publish quite well, but that's not so interesting. I think the sweet spot is where you try to articulate general principles with their implementation and of course, computational work. It helps do this bridge. And that's just a summary. I told you about the worm, but I've worked on the larva, the sweet larva, mice, and lately we're doing things in humans, kids, and also robots. And I leave it there for whatever discussion. Well, this is just like an image of the renormalization group in physics, hardcore mathematics. But you see, here you have the worms. You would make groups and groups of groups. These are very powerful concepts in physics. Let me end with this. There's this struggle of theoretical physicists working in biology. We try to copy and paste all these amazing conceptual tools. Most of the time, they don't work. So my job is trying to see which ones work, and if some don't work, to be honest and ditch them and say, no, biology is not a new physics, like trying to force, well, I'm not going to say theories because I respect them, but you see my point. Okay, so I leave it here. Thank you.
Thank you, Alex. We're open for questions. So,
I have a question, maybe. Hi, Alex. So thank you for the really nice talk. And so I have a maybe a discussion point. So as you mentioned, the choice of partition usually is not blind for science, for experimenters. So we do the partition because, or it's related to what we want to understand, or we have some sorts of evidence. So you use the mutual information as the measure, and I know that you could use some other type of measure. But in principle, I would say that for example, when I'm studying calls, right? So there is a reason for me to divide, for example, the calls in type of calls, right? Or either through the acoustics, or either because I know what kind of behavioral response would cause. So my point is that even though it might not be optimal from the predictive point of view, still could give a lot of information about the behavior. Right?
Yes.
So my point is that maybe part of the problem is also what you should use as a measure of like a goodness. So in your case, as you said, you use mutual information, but if you use other type of criteria, probably you would get some very different type of conclusion. No?
Yeah, totally. Here we used mutual information interpreted as predictive power, because it's all these information theory measures sound kind of God-given in a way. So you know, but I agree. I could have decided another criteria that it's not predictability. Also because we wanted to relate it to the issue of replicability. But yes, you could decide other criteria. So that's open. But I think there are 3 points here. One is you say you decide your partitions, but I'm not sure how often we are aware that we are deciding or what we're really deciding. Every experimenter or person that analyzes data knows that they more or less decide something, but probably not really what they decide. What will be the consequences of maybe deciding totally different or slightly different, as I tried to illustrate here. You could use totally different or just make a little mutation. And also, and that's the question that I was more speculative at the end, whether we may choose whatever we want, again, predictability, but do we care what's the right, I'm not sure if that's even a right question, but what's the right partition from the perspective of the animal? At least it's worthwhile asking. I mean, there are many people insisting on this, like recently this book by Buzaki, right? The Brain from the Inside Out. He's also making this point, right? Like sure, the ideal observer from the outside may have their own interests. We have our interests as scientists, again, conscious or not so consciously, but even then, it would be worthwhile asking, well, from the perspective of that organism, what's the best partition? And if we could find that, I think we'll be doing true ethological work, and then neuroscience would benefit a lot. So I think that's the chain of reasoning. To be better neuroscientists, it's important to be more organism-centric.
Yeah. So I totally agree with you. But let's say, but again, even when you do some record, let's say that I use some Kinect to take a video and do a partition that is animal-centric. Let's say that I try to minimize my criteria to do partition and not try to use something that maybe the animal you use to partition, maybe just by some using some algorithm or I don't know, some other way. But in any case, the point is that whenever you choose, for example, to use a video or whatever, you already put partitioning the entire possibility of observable. So my point is that it seems to be a problem that you cannot escape in any way, right?
Yeah, I see. I was understanding you. Yes, I mean, this helps me make another kind of conceptual point that turns any physicist on, which is that maybe it sounds too esoteric, but that the mind of the observer cannot be totally separated from the observed phenomena. And I think I'm not invoking quantum coherence in microtubules here. Who knows? I'm saying even when we do that, even for the tent map, not just the worm. Our theories of life, let me put it more poetically, our theories of life, I think, are entangled with our theories of knowledge. And I think that's often not considered also in neuroscience, because we think, you even hear like this is they say an unbiased approach. Well, it's not an unbiased approach, it may be unsupervised, it may be using some very powerful concepts, but everything is biased in a way. We're always projecting in a way. So again, as a philosophical, theoretical point, I think it's worthwhile to remember it from time to time. Yeah, but I agree.
Thank you.
Yeah, thanks, Dan.
I have a question about the partitioning idea that you talked about. Oh, and congrats by the talk. It was super nice, by the way.
Thank you, Tiago.
So my intuition is that from the way that you're measuring how good the partitions are, basically the partitions that are highly correlated among itself, like when the elements of a partition are highly correlated with themselves and very not well correlated with the other partitions, this would give a good prediction. Is that right? Is that intuition correct?
Yes, well, you should make more concrete what you mean by correlated, but yes, there's intuition there. Like if the worm is in this posture, and it's very likely, and it could be for different reasons. One could be postural, the morphological similarity. I mean, if you're like this, well, I'm not sure if you're seeing my screen, but if you're like this next, and the time resolution is good, it's fine. You're not going to be like that in the next frame. So sure, there's something that's constrained by the body, and there's also that probably is constrained by the sort of neural commands that the worm has evolved to have this lovely elegance and sinusoidal movement. So there are all these types of, I don't know if we call it correlations, but sure, where you are and where you're going to be, I would say more constraints actually. Where you are and where you're going to go is constrained by several things. But the question is, out of those constraints, what's the best way of making, in this case, partition, so that you can capture another way of putting it, so that you can capture. You're gonna throw data. You're gonna throw details. That's for sure. So which details are worth keeping and which details are worth throwing away? it's a more pragmatic way of seeing it, right? So this is a way of saying, but Dan was suggesting that maybe there are other ways, but this is a way of saying, well, I can throw out these details and still capture the intrinsic variability of what's going on, given the constraints.
Mm-hmm. And on that same vein of throwing away details, despite having a potentially higher predictive power within the macro partitions, is it true that if you're using them to reconstruct the behavior, you'll lose some accuracy?
Sure, of course. So if we go back to the first kind of little story I told you, we have these 90 templates. So the way you estimate mutual replaceability is that you have these 90 templates, and you build some sort of a Markov transition matrix. So there you're already throwing a lot because you're not going to look beyond. But then you ask, how am I going to order these postures so that this analogy of the menu takes place? Basically, which ones co-occur? I'm going to put together those postures that co-occur with elements in the future and the past, not with themselves, right? And that's how I'm getting that already there. When we made an analysis, if you look at Saurabh's paper, the context-free grammar for C elegans, there you can look at the eigenvalues and see how much you're losing information with the Markov transition metric. But sure, if you use a Markov at some point, you're already destroying structure. By the way, you can also calculate. I didn't show it. It's down here, but it's not worth in any case. And this predictive power, you can also ask not just the immediate next point in time, but further. So you can scan it. You can scan. Well, I'll show it. I have it here. Is my screen shared still?
Yes.
Yeah. So you see, you can jump. I mean, it's not a way to. You don't recover what you lost completely, but you can also ask what happens if I scan further. Right? Yeah. And this could be also a way of looking at a Markovian, because you're asking initial and next one, right? But sure, there are more sophisticated analyses that don't rely on a Markov chain. Yeah.
Nice.
Fantastic. So, and I'm super naive. I'm not a mathematician, and a lot of the math behind it, I don't really understand. So when you calculate these transition probabilities and you group them, can you estimate between those meso states, what are the transition probabilities between them, and use that as a predictive
well,
tool?
So that would be like doing further analysis or further modeling on your data, right? So, well, in a way, it's what we did. Let's see. I'm not sure if that's what you're asking, but let me just jump to here. So that's the story of the mutual replaceability, where we start with this 90 and we make 10 groups. Well, they are made by our criteria of mutual replaceability, 10 groups and 3 groups. Now, if instead of, I didn't show you here the transition matrix of 90 to 90 because it's quite sparse and so on. But if now you look at the transition matrix of 10 to when you have 10 modules, then that's the point precisely. I think that's what you're saying. You get a lot of predictive power because you're grouping things in a way that you capture this roaming structure of the worm. Whereas if you would shuffle, this is like you would take these 10 groups and you would randomly shuffle the postures you put in them. Of course, shuffles are always like help you make the point because they work really bad unless there's something weird with how you did things. But if you would shuffle the group, the microscopic postures you put in the 10 modules, then your predictability is really low. And here you can really know with a lot of certainty that if you are in subgroup B1, you'll be in B2 next. Now, which posture of B2? Well, you don't know, and maybe it's good not to know. By the way, this is another important thing. We want to predict animal behavior so well, but there's a limit because animals also, because they cannot be infinitely precise in their motor control, but also because they may want to generate variability, they don't always do the same thing in the same way. So you see again this trade-off between what we would like to and what the animal can or wants to do. Yeah.
Fantastic.
May I ask a question?
Of course.
Alex, I was wondering about your data set. Is your data set grouped by environmental circumstance or states, or
Yes,
you have
so
all
this
these
goes,
behaviors, the sequence of behaviors that you have are treated as if the animals had all kinds of experience in the
yes,
controlled environment that you have, or
yes,
you can also apply that for groups of particular environmental
yeah,
states that
yeah.
you could have flips of your groups, your
Yes, sure,
partitions
sure.
in the
So in the
beginning.
same way, we always do this. There's always figure whatever 6b, where you explore different environmental conditions, and there's always figure 7 where you explore different mutants, right? It's the difference game, I call it, a bit ironically. It's important, but what interests me is more the concept. But sure, I mean, this data set, this lovely data set, has, as I was telling you, more than 1,000 individuals, like the wild type, and then all of these mutants and wild strains, and they are tested in on-food condition and off-food conditions. So if you imagine here, you could have C. elegans scrolling and with no food, or you can enrich it with bacteria. I think that's what they like to eat, right? So and of course behaviors are different. Like by the way, each of these individual tracks is for 15 minutes, I think, because otherwise you don't control somewhere for stationary behavior. So what does that mean is that if they're very hungry and there's no food around, they will be exploring more to find it, maybe doing levy flights or whatever. But if they're already satiated, they will do this less, and so on. And of course, in these manuscripts here, I think we show it, and in paper with Saurabh, I think we also show it, what happens when you change, when you do the same thing, but the condition is different. And I mean, that's why I say it's important, but at the same time, if you change conditions, things should change, right? Again, what's the theory behind? You see, I don't want to make fun of that, but we could check, let's say, you could say, let's see what happens when we test the worm on food or off food. And if predictability goes up, you'll post hoc make sense of it. Sure, I mean, it's more predictable because when there's no food, it's going for it. So there are more long runs, and therefore locomotion is more stereotypical and it's more predictable. But if it's the other way around, you'll also find a narrative for it, right? So I'm saying that it's worth as an exploratory tool, also to realize that the things you find make sense, like what we did here when we were compressing the motifs. These are generated automatically, but then you also need to look at them carefully and see if they are biologically meaningful. And if they are, you're more on safe ground. And then what you can do is try to fish some weird ones that you would not expect biologically. So maybe that's how you make a bit of a business with these computational tools.
Right. Because my question is, at the long run, at the equilibrium that you get of this transition matrix in the long run, are you actually getting a sense of the physical constraints that you have of behavioral sequence in general? Are in at the same time you're excluding, or based on the model that you use to make the predictions that you can make, and you're leaving aside, for example, stressful or conditions where the animal point of view is kind of having a lower weight in this sense, because if you put in very discrete states, you have this condition with food, condition without food. If you clump this data together and you run your predictions, you can have some. But if you test the environment and you put the biology inside the model, that say the environment is constantly changing and you're creating a very stable state for the animal. I'm just trying to put biology inside that, and trying to check in a sense that the environment can change quickly, and do these predictions keep the same in these conditions? Would that be able to test, or have you tested that?
I see what you mean. I mean, we don't lump together worms that are on food and off food conditions, because that would create confounds, and And so you do it separately. But I think what you're saying is, if I understand correctly, what we learn from this in one condition or the other, can we generalize and learn something general about these two, right?
Yes.
Is this what you're saying? And you're asking about the model, but there's not really it's a behavioral description. There's not really a model in the sense of, we're not describing the worm as a Markov oscillate or as a chain or something like that. It's more like a behavioral description that's refined enough, but also has it's throwing away some data because it's discrete in space and time. Put the biology in it. When you say putting the biology, sometimes people mean mechanism, but already, if you think in terms of ethology, already looking at the structure of the worm, detecting the structure of the worm with these methods, I think it's already revealing part of the biology. But yes, let's move to this slide maybe to illustrate. Like this is what we propose as these grammatical rules that we detail much more in this manuscript. But the question would be, well, but how does that is change, or how is this modulated if the worm is hungry, if it's not hungry, if it's time to mate, etc. Right?
Right.
And we are not there yet in the sense, because that would be
19 to a different group depending on the conditions. Or,
yeah, I see.
As you said, they could make a very weird body flip, but in one condition, but not in the other.
Yes.
I was just wondering about that.
I see. No, and for instance, what we do, we extract these groups for N2 worms, the wild type, and then we test how we could explain the wild isolates, for instance, and it works pretty well. So maybe this is another way of answering your question, right? You would derive it for a certain condition and then test if it makes sense for other conditions. And if it more or less holds, it means that it's, you would want, in this case, you would want the groups and the to be the same, but maybe the probability of transitioning in these syntactic rules to be modulated by that. So that's what you would want, right? Because if the groups change, if everything changes, then you need to make it from scratch every time. And so you're learning something, but you're not learning something very general. Also, relationship between roaming and dwelling. If you check our paper, right, we also have some rules for dwelling, but how upper up you would decide to roam or to dwell, that we don't know, right? So you would want it more general, yeah.
Thanks.
Yeah. I would ask you a question, anyone in the audience, which is, what's the best attempt you know about what I'm trying to call a theory of measurement in behavior, but if nothing comes up to your mind in behavior, in theoretically inclined, like here you have the phenomena, and here there are some theoretical principles, like Newton's laws, but again, I don't want to be too physicist-centric. Like, this is what you should measure, and that's why you should measure this and not these other things. What's out there that resembles that?
I would say, like, if they very in a broad sense, where and I'm pretty sure that you thought in a similar kind of thing. But if you think about the Umwelt idea and the functional cycle, right? I feel that what you should measure are the things that compose the entire functional cycle to form the Umwelt. So if a biology wants to understand the behavior, I think you would like to understand the functional cycle as described by von Uxkull.
Yes, I see. But that changes animal by animal, right? Species by species.
Yes.
Yes.
But still, you could. I think that you could have a theory, like, let's say, as you said. So you have foraging, right? So you could actually compare different species by describing the functional cycle for different species, but probably there could be something in common for all these different species. Right?
I see.
I want to jump in this as thinking about, like, what one should measure, and I was thinking, like, probably Alex, Daniel, and I we're both, I mean, these days interested in how to tackle spontaneous behavior, naturalistic behavior, but probably because we come from a neurological interest, right? Like, how does the brain orchestrate behavior? Maybe I'm wrong, but I suspect that we are not in ethology for ethology, that we are in ethology for thinking about neurophysiology, right? And what I was thinking was, when deciding what to measure, one also should be thinking of what's going to help me the most understand how the brain, brain body, but how this system, including the brain, is implementing this. And I'm thinking that, couldn't there be a problem if you're applying conceptually this mutually replaceable elements thinking, and if you apply it to the extreme where you lump together the mutually replaceable elements, couldn't you be heading into trouble when you're when then you're going after how the neural systems are implementing this? I was thinking about your example of your code, so you could either turn on the heat or put on a coat. If you lump those together and forget about having lumped them together, you could be heading into trouble, because I'm sure your brain is solving very different problems when you put on a coat or when you turn on the heat.
Well, maybe at the level of motor implementation, it's very different, but at the level of high-level goals, it's trying to solve the same high-level goal.
Like, that's a small component of what's actually going on. It's only a part of what's actually going on. The implementation, because also then once you go into the implementation, you have a lot of like sub goals that the system is solving, like how you get off the chair, how you reach whatever you're trying to reach. So I'm saying, just in general, that if you take the what would it be, MRE to the extreme, I think that could cause some problems in the neuro side of the story.
Yeah,
I have actually this could be so I don't know if we should continue. Tarcísio, you should decide it, because I don't know at this point discussion seems to be fine.
No,
okay, so.
No, absolutely, we should continue.
So the way that I see it, actually, it's a little bit different. I maybe in this sense I agree more with what Alex said, because, like, for example, you could I think that is related to how you interpret what is the function of the brain. Right? So if you feel that if the activity in the brain are different, it means that the brain is doing different things. I agree with what Diego said, but I don't think that that's the case. Like, it's totally possible that the brain activity is totally different, but in some sense is doing the same thing. So let's go more to the Alex side. So if you think, like, for example, I can think about physiological system in general, right? Like when you see hearts, right? So the function of the heart is to pump the blood, so that actually the body will have enough oxygen, right, in some sense. So if you think in this way, actually, from the functional point of view, every change that happen in the heart, like increase the heart rate or decrease or whatever, right? It's just way to achieve this aim in the end. So in this sense, I think that actually you get a different type of understanding. If you go in the heart-centric point of view, you would say, if it's pumping more or pumping less blood, it's doing different things, right? But maybe actually they're doing exactly the same thing, which is just to give enough oxygen supply to the body, right? So I feel that's exactly the point, right? The Alex has made.
Also could be illustrated here. Let's see if some of these, you can have, let's see, you could have, this is the multiple realizability problem, right? You can have not just different structures that accomplish the same function, but you could have maybe, and for instance, Paul Katz has shown this in very simple organisms. Well, maybe he doesn't like that I call them simple, but the point is that you could have even the same neural commands and everything, and slightly different body, and then the very different behavior. or the opposite, right? You could have very different neural commands that lead to the same behavior. So I think we must not forget that these are different levels, and the mapping between the levels is not one-to-one. So yes, it would be nice to expect same neural activity, but even if it was different and would lead to the same behavior, it was the same and it would lead to different behaviors, we could make sense of that if we would integrate, again, what the brain does in a vat with the body of the animal and where it is in the world. I think by integrating this, then we can navigate this challenge of biology, which is that the levels are not, you cannot map nicely from one to the other. So everything bifurcates all the way in space, like we try to explain here, and also in time, right? To make things even harder. Yeah, animals are constantly learning and developing.
Okay, maybe what I'm saying is that I mean, one should, if you go for, I mean, I like the multiple replaceable elements approach, right? But I'm saying it's like, if you go that way, it's like, I think at some point you should remember what's inside each grouping that you made, right? And understand that there could be variability inside, and it's not just like continuous variability. There could be like these distinct states that map to the distinct alternatives for the multiple replaceable element.
It's complicated. I could raise my hand to call a taxi, or to just, or I'm serving a ball to play tennis, and maybe my neural, if you record somewhere, the neural subset, the neural activity will be the same, but maybe somewhere else it will be very different, because that's what knows. I don't want to play the neural homunculus here, but that's what knows I'm playing tennis or I'm calling a taxi, right? So again, one needs to zoom in and zoom out in space, and also in time and in abstraction level, right? I decide I go to work, and then I can decide I'm going with motorbike or with my car, or maybe take and each of those brings me into a new module where I now need to implement, okay, if I want my bike, I need to remember where the helmet is and then put it on. But if I go by car, I don't need to do this thing physically, right? So different levels of organization, of analysis, of abstraction need to be combined. Yeah, it's difficult. And concretely, in the case of the worm, yeah, I see what you mean. I could be detecting these mutually replaceable elements, and I could be then, by doing that, not paying attention to connections that are not going to happen immediately at that window of time, and maybe are more relevant, or the other way around.
Alex, in a kind of a related but not related question. So basically, I think you mentioned a little bit. So in the case of the worm, the kind of behavior that you're measuring, there's several physical constraints and perhaps neuronal constraints in terms of what kind of body position and transition probabilities you're going to have in different states. And you can measure that, and you can group with this. Do you think we could expand for your type of modeling to other types of behaviors that are not constrained by, muscle or physics and body?
Yeah. No, that's a great question. As I said, the worm is useful because it's mostly 2D, because how it changes body posture is very tightly linked to where it's going to go, and so on. Also because, as you know, the worm has over 300 neurons, so potentially you could start mapping everything, despite the great promises and relative failures of the connectome and so on for the worm. That's another discussion. Now, then I think that the powerful thing about, and I have it here, treating behavioral data as text, and then running all these computational analysis to extract structure, be it in terms of syntax or without syntax, making partitions and so on, is that it's like you're studying a language of an ancient civilization, or you come from outer space to the Earth, in the sense that, well, give me a sequence of symbols. You can choose your alphabet. It could be short, it could be long, I don't need to know anything about semantics, although, of course, and that's maybe related to the question of the biology, I would want to bring semantics, because ultimately my goal as a neuroethologist is to understand how the animal is solving its problems, right? But I could play we're in YouTube, I'll say this kind of pornographic approach where I say, I don't care about what's going on here. It's just mechanical. Give me this list of symbols and I squeeze it in these many ways. And I'll try to propose to you the structure that it reveals, which is what you could do with ancient texts. If maybe you don't have a dictionary, right? Or you didn't even know how to put the letters together. Like as I was trying to illustrate, where was it? Here, right? Well, you would do this, we run it on text, and you detect is many times. Maybe this is a hypothesis for a next experiment. Why is this weird behavior having is all the time? And look at these long behaviors here, and that could help you. So I think, to answer your question, it's ready to try other behavioral data that makes sense to describe it symbolically. Although I'm a great fan also of kinematic descriptions, positions, velocity, orientation, and reading that as if it was music with all the degrees of freedom. And really, by the way, that other way of looking at behavior, I think it helps you a lot to be in the mind of the animal, because you're imagining yourself moving, stopping, rotating. It helps you with this Untitled problem I'm obsessed with. But this is if you have a lot of data and it's precise, this is also very, very convenient to try to reveal a structure that then you need to interpret again.
I'm sorry, fantastic. So does anybody have any other questions? If not, I would like to thank you, Alex, so much for speaking to us. It was a fantastic talk. I certainly learned a lot. I'm actually going to go back and read more of your papers because now I'm more and more curious about it. And I really appreciate your time.
Obrigado. Próxima vez fazemos na sua língua. Eu adoro.
Em português. Perfeito.
Força com o confinamento. Vamos a volver malucos aqui,
Sim. Nós estamos há quase 6 meses. Então...
Boa. Sim.
Ótimo.
Eu mudei. Eu estava a viver num piso pequenino. Mudei para... com as pequeninas é duro. Mas todo mundo está a sofrer de alguma forma. Então é bom fazer estas coisas virtuais. E depois voltar ao mundo real assim.
De máscara. De novo, muito obrigado, Alex. Prazer conhecê-lo. E espero, de repente, em breve, a gente consiga fazer algo não virtual, presencial, em algum momento.
Great. Ok.
Ótimo.
Tchau.
Thank
you. Take care.
Diego?
This virtual world. No me gusta mucho.
No.
This is real. It's just like across the internet. But...
Ok, até já.
Até já, obrigado.